Morning, ladies and gentlemen, and welcome to the 14th SAIMM webinar. I'm Buhle Xakalash, a Chief Engineer in the Pyrometallurgy Division at Mintek, and I'll be chairing this webinar as a follow-up to part 1, which took place earlier in the week, presented by Tamsyn Grewar, who was assisted by Kerri during the Q&A session. Both ladies are senior scientists in the Biotechnology Division at Mintek. Tamsyn gave an overview of the severity and complexity of water-related challenges in South Africa, and introduced to the audience to a treatment technology that the Mintek's Biotechnology Division has been developing, named cloSURE. Today, the seats will be switched. Kerri will present to us, and Tamsyn will offer an extra hand during the Q&A session at the end. To recap on Kerri's bio for those who missed Tuesday's session, she is a senior scientist in the Biotechnology Division at Mintek, as mentioned earlier, and she graduated with an MSc in engineering from the University of the Witwatersrand, and has accumulated six years of experience in environmental management, as well as 6 years of experience in anaerobic digestion and wastewater research. Kerri is currently working on Mintek's flagship research project on biological treatment of acid mine drainage, and has presented at a number of local and international conferences. And looking at this bio, ladies and gentlemen, we are certainly in good hands. And back to today's topic, the ladies will be taking us through part 2, which is developing the solution for the cloSURE process, the story of Mintek's passive sulphate reduction process for treatment of water after mine closure. For your information, ladies and gentlemen, the presentation will be circulated to the participants. And please kindly ask your questions on the chat under Q&A. And we have exactly 1 hour then for the entire session. And over to you, Kerri, and looking forward to another exciting presentation. Thanks.
Thank you for joining us today for the second part of our series about Mintek's cloSURE technology for the treatment of acid mine drainage for mines post-closure. During this presentation, I'd first like to highlight the extent of the acid mine drainage problem and what is being done about treatment. And then I'll tell you a little bit about biological processes, those ones that our technology is based on. And I'll give you an overview of the development of the process and the results that we have obtained. Finally, I will share some of the exciting project development work that is happening and tell you what comes next. So this river is severely contaminated with acid mine drainage. You can see the red color, which is from high levels of iron in the water. You will notice that this ecosystem is completely devoid of life, and there is no riparian or aquatic vegetation anywhere to be seen, and there are no aquatic animals in the river. And this is because of the chemistry of the acid mine drainage. The pH is typically low. It's between 2 and 6, and the water that we are working with originates from the coal mines in Mpumalanga, and it's typically between a pH of 2 and 3. The water also contains high concentrations of sulphate. South African waters contain uniquely high levels of sulphate in excess of 3 grams per liter, and this increases the salinity of the receiving water and has significant impacts on human and livestock health, including digestive and respiratory issues. Finally, the water contains heavy metals in concentrations that are high enough to pose a severe threat to receiving water users, but too low to feasibly recover. Damage costs of sulphate pollution on river systems are estimated to be around 7.7 million rand annually. The true cost of acid mine drainage is unquantifiable, however, in terms of community health and environmental degradation. So where is all this acid mine drainage coming from? This map is from the Mine Water Atlas, and it uses GIS data to show the mineral provinces across the country, which are these pink regions that you can see over here, as well as all of the mining projects for South Africa's major commodities, which are sort of aggregated over here. Acid mine drainage typically originates from coal and gold mining activities. So I'd like to draw your attention to the gold reefs over here. And these yellow dots are all of the gold mining projects in this region, and to the gray dots over here, which are the coal mines. And you can see that most of this is in the densely populated areas near Gauteng and Western and Mpumalanga. So the coal fields are estimated to generate around 250 megaliters of acid mine drainage per day, and the gold mining areas are estimated to produce around 350 megaliters of acid mine drainage a day. Now, these volumes are staggering. So government have what they call a short-term plan, which is the construction of 3 high-density sludge plants, which will neutralize acid mine drainage from the eastern, central, and western basins, where all of those yellow gold mines were in the previous map. Now, CapEx cost to date total around 2.6 billion rand. So this picture on the slide is a picture of the Eastern Basin Acid Mine Drainage Treatment Plant, which is located in Springs. And it's exciting because this is the largest acid mine drainage treatment facility in the world. But there are no centralized HDS options available or planned for the Olifants Catchment. The government's long-term plan involves an investment of around 8 billion rand for the construction of desalination plants like this one in the picture to treat neutralized acid mine drainage and to produce water for discharge or for domestic use. The energy requirements for reverse osmosis are high, and the process is one of the highest treatment costs per cubic meter, and it can produce large volumes, and sulfate concentrations can be in excess of 9 grams per liter. So let me return for a moment to the Olifants Catchment, since our piloting and demonstration activities for the closure technology are going to be based in the Witbank coal fields. So, which are over there. All or most of the Witbank, Eastern Transvaal, South Rand, and Gaangwane coal fields are located here in the upper catchment. The Olifants Catchment is of national importance because it has the second largest irrigation scheme in South Africa, and this area is of high agricultural importance to the country. It also has 9 major coal-fired power stations and 800 mines in the region. There are 65 collieries in the upper Olifants region alone, and all of these mines have diverse characteristics, producing different qualities of acid mine drainage. So this map from the Mine Water Atlas really drives home the scale of the impact in this water management area. The map indicates where the highest risk of production of acid mine drainage is within the catchment. So these red areas over here delineate the areas where there is a very high risk, and these yellow areas show where there is a high risk of acid mine drainage being generated. It is estimated that approximately 170 megaliters of acid mine drainage per day is released into the upper Olifants Catchment. Here, there are no plans for high-rate HDS processes or desalination plants. So most of the large coal mines neutralize their acid mine drainage prior to discharge using lime or caustic soda. And while it is better than no treatment, the problem of sulphate and metals contamination remains. And where they cannot discharge, they store the water on site in storage dams or evaporation ponds indefinitely. So we need alternative options to protect this important catchment area and to reduce the health and environmental liabilities of the mines. So I'd like to introduce you to biological sulfate reduction. It's another mechanism that's used around the world to treat water containing sulfate, and it's a good potential solution for treatment of point sources of acid mine drainage in the Mpumalanga region. It's typically used for recovery of metals and for production of sulfuric acid. And it's driven by these sulfate-reducing microorganisms. These microbes require a carbon source or an electron donor for biochemical processes for energy production. So biological processes require the addition of an organic substrate. Typical substrates include simple organic compounds, predominantly short-chain compounds such as lactate and acetate, but also ethanol and hydrogen, depending on the microbes present. This is a major cost for operation of these processes and is the largest barrier to implementation of the process at scale. So we also have active and passive processes, and Sulphitech is a high-rate active process, and it's just one example of a process that employs sulfate-reducing bacteria to reduce sulfur and precipitate metals. And on the other end of the process spectrum, we have passive biological processes. And these have a number of advantages. They have much lower costs. They have low to no energy requirements depending on the process, and they rely on complex substrates which are degraded over time to reduce operational costs. Anaerobic bioprocess is also robust, and once at steady state, they can tolerate variable hydraulic flows, variable water chemistry, and operating conditions. Again, around the world, The aim is to promote enough sulphate reduction to remove metals prior to discharge. So you can see how a passive process like this would be suitable for smaller point sources in the remote locations of the Olifants Catchment. Just some examples of what they do in other regions. In some cases, they have acid mine drainage discharges into pit lakes. And what's a common strategy is to inject ethanol into a pit lake to promote sulphate reduction in the lower anoxic regions of the lake. And these remove the metals from any discharge streams from the pit lakes. Another example is a permeable reactive barrier. And this one is constructed around a mine dump. And what it does is it treats runoff coming from the mine dumps to remove any excess heavy metals and prevent contamination of the receiving environment. So in South Africa, there are 5 examples of biological sulphate reduction plants. BioSure and Vitasoft are active processes, and this is a picture of the BioSure process. And I won't go too much into them because they are active. Anglo American operates a bio-neutralization plant in KwaZulu-Natal. The aim is to employ sulphate reduction to increase the pH and remove metals from acid mine drainage prior to discharge. This plant is still in operation after 5 years. There was also a pilot-scale semi-passive biological sulphate reduction system that was operated for 2 years at the New Vaal Colliery in Middelburg. And this is a picture of that pilot plant. And finally, the 5th plant in South Africa is Mintek's pilot plant, which I will discuss in a moment. The system is different in that the aim of the process is to produce water that is fit for use for irrigation. It's designed to remove high concentrations of sulphate in excess of 3 grams per liter. And to feed our sulphate-reducing bacteria, we use waste materials, which we use as the organic substrates, such as cow manure, hay, food waste, and crude glycerol, thereby overcoming the inhibitory costs of substrate. The process will ultimately be integrated with irrigated agriculture to drive microeconomies after mine closure. So let me give you an overview of our closure technology. Closure has been developed as a low-cost, low-maintenance technology for treating acid mine drainage and is particularly suitable for treating small point sources in remote locations that lack services and infrastructure, such as mines post-closure. I'll tell you in a moment how we successfully piloted this technology, and we produced fit-for-use water. And it aims to become part of the mining industry's long-term rehabilitation strategy post-gold or coal mine closure. So this is a schematic of the high-level concept of our treatment process. So we have acid mine drainage that is acidic, it has high sulfate levels, and it has metals. And as we mentioned, acid mine drainage tends to be a severe and persistent problem in the environment. So the first stage involves pre-neutralization of the acid mine drainage, and we employ biological sulfate reduction to remove sulfate, increase pH, and remove the metals in the water. This is the 3-pronged approach mentioned by Tamsyn on Tuesday. Reduction of sulfate results in sulphide, which can be corrosive to irrigation equipment, and it can pose a safety risk to plant operators. So in the second stage, the focus is on biological removal of sulphide and the recovery of sulfur from the water. In the lab, we've removed more than 85% of the sulphide, and we've actually been able to produce irrigation quality water. So the intention is to integrate the treatment process with irrigated agriculture for the production of value agricultural products. The long-term goal is to promote agri-industrial hubs, which will provide employment and food security to the country. So this is the biological sulfur cycle. This cycle is vital because all living organisms require sulfur to manufacture amino acids and proteins. So similar to the carbon and nitrogen cycles, sulfur species are utilized and converted and then shunted around the ecosystem. So in designing a process to remove sulfate and recover sulfur from mine water, 2 parts of this cycle are of interest to us. Firstly, dissimilatory sulfate reduction, which is done by sulfate-reducing bacteria. You will notice that there are 2 pathways here. One is assimilatory sulfate reduction. And what happens here is the sulfate is consumed by the microbes and it's used to produce more biomass. So it's incorporated into the cells. And this sulfate reduction doesn't use a lot of sulfate in a receiving water in a treatment process. So we rely more on dissimilatory sulfate reduction. And what this means is that the sulfate is taken up and used in the biochemical pathways for anaerobic respiration and is converted to hydrogen sulfide. These bacteria and this biological sulfate reduction pathway are harnessed in the first stage of our process. Secondly, we have sulfur oxidizing bacteria that return the highly reduced sulfide back to sulfur and sometimes even sulfate. These bacteria form the backbone of the second stage of the closure process. So let me go into a little bit more detail. For any microbiology nerds watching, here are some electron micrographs of sulfate-reducing bacteria. I think they're beautiful. These guys are quite amazing bugs, and they do most of the heavy lifting during the water treatment process. They're a really diverse group of microorganisms, and they're found among the prokaryote domains of archaea and bacteria. They were the first organisms to evolve on this planet 3.5 billion years ago, before there was oxygen in the atmosphere. So they are obligate anaerobes, and this means they cannot tolerate any oxygen. They use sulfate instead of oxygen during respiration, and they require an organic carbon source for energy metabolism. And they have an optimal pH on average of about 5.5. And they're found everywhere, in the digestive tracts of ruminating animals such as cows, in sulfur springs, and in soil and aquatic sediment. And now, with high levels of sulfate present in acid mine drainage, they've also been found in streams of decant where the environment is suitable for their growth. At Mintek, we've isolated sulfate-reducing bacteria from cows, from mud, such as in that picture, and also from areas of acid mine drainage decant. So how does it work? First, we need to pre-neutralize the water so that we increase the pH to around 5. Now, I know you're thinking, but you just mentioned that lime neutralization is expensive and that it produces hazardous waste, which is true. But we use less lime as we are only reducing the pH enough for an optimal environment for the microbes, and we're not aiming for precipitation of any metals. So due to the additional waste stream that lime neutralization produces, our closure team is already on the problem. And we have a few lines of research at Mintek looking into alternatives such as bio-neutralization. Now, the main tank will be a rectangular pond with a packed bed or substrate to immobilize the microbes. They form biofilms on the substrate. Once it's steady state, the bed will house a complex consortium of these microbes, and that complex consortium will include sulfate-reducing bacteria. So our tank or our pond has a downflow configuration to prevent escape of sulfide gas and to ensure higher concentrations of sulfide for oxidation. While the bed might liberate some organic substrate for the microbes, organic material needs to be fed to the organisms in the bed. As I mentioned, the cost of substrate is the largest barrier to implementation of the process at scale. So we've investigated the use of waste materials to provide the electron donor to the system, such as wood chips, cow manure, food waste, and waste glycerol. And we've actually had quite a lot of success. So we've also investigated the use of waste plastic as part or all of the packed bed. And a number of strategies are currently under investigation at Mintek to optimize substrate addition and delivery, as well as process adjustments to maximize the techno-economic potential of the integrated process. So watch this space. During startup, the bed is colonized by a diverse consortium of microbes. The microbes break down the available organic material into simple compounds such as volatile fatty acids, those lactates and acetates I was talking about. And these are the preferred substrate of the sulfate-reducing bacteria. Using acetate or lactate for energy, which is denoted here using the generalized term for organic substrate, and using sulfate for respiration, sulfide and bicarbonates are produced in the system. And this drives up the pH of the water and it produces alkalinity. Sulfates are also removed, and sulfide, the sulfide produced binds to any metals in solution to form a metal precipitate, which is retained as a stable solid in the system. Our division at Mintek has been developing the process for the past 8 years, and we started with sulfate reduction in modified 2-liter BioLeach reactors, if you can believe it. And 3 years ago, we built a pilot plant at a coal mine in Mpumalanga, processing 250 liters per day. This was designed to be a 3-stage process, but our results were better than we imagined, and it turned out that all of the sulfate was reduced in this first reactor. So I'd just like to show you the sulfate results from the pilot plant, because this gives an indication of just how well the system was working. This black line shows the sulfate concentrations in the acid mine drainage that was fed to the plant, and this purple line shows the concentrations in the treated water. So this initial drop in sulphate concentrations shows that the plant reached steady state around day 75, when low sulphate concentrations were measured in the treated water and high levels of sulphate reduction were achieved in the reactor. So this was a period, there was a period here between day 350 and 400, where more than 99% of the sulphate was removed in the water. So you can imagine the incredible concentrations of sulphide that were produced, which at times reached 880 milligrams per liter. Another point to note about this data is that the feed concentrations of sulphate were highly variable, typically between 2.1 and 4.5 grams per liter. And while the sulphate concentrations in the effluent were actually fairly constant, anaerobic processes at steady state can handle variable feed conditions in ways that aerobic processes don't. Toward the end of our piloting activities over here, you can see that sulphate reduction slowed, and this was likely due to bed degradation and hydraulic short-circuiting. But we didn't hit the crash phase as early as expected, probably because we were manually adding organic materials as a substrate for the microbes. So the blue line on the graph is the temperature inside the reactor, and it shows that our sulphate reduction rates were stable through a summer and a winter cycle. So in all, this piloting project was a huge success. But there was still work to be done. So while we were piloting in Mpumalanga, we were still developing the second stage of the process at Mintek to polish the effluent and achieve fit-for-use water. While we were able to treat the water by removing sulfates, increasing pH, and removing metals, we were just short of irrigation quality for some of the parameters. The treated water contains high concentrations of sulfide, and at neutral pH, around half of the sulfide is present as a dissolved gas. This poses a health and safety risk, but it is also corrosive to pipelines and irrigation equipment. And we also had a number of metals that exceeded the water quality targets for irrigation. So in stage 2, we use sulfur oxidizing bacteria, and these are found in the natural environment as well, also mostly in aquatic sediments. They often coexist with the sulfate reducers that we saw in the previous stage. In some cases, they form these mats that you can see in these two pictures over here, at or near the water surface. In this picture, this is a mat of purple sulfur oxidizing bacteria. And in the second picture, you can see the presence of sulfur in this mat. Now, if we were to zoom in and have a look at these beautiful electron micrographs, you can see that sulfur granules actually accumulate inside the bacterial cell. So again, these are a highly diverse group of microbes. But the key things that we need to know in developing a process is that sulfur oxidizing bacteria can be aerobic, anaerobic, or facultative, which means they can live in environments with varying levels of oxygen. The ones that we are interested in for our process fix carbon dioxide as a source of carbon, and they also metabolize sulfide and accumulate elemental sulfur inside the cell. If sulfide concentrations are low or they become depleted, they then oxidize the sulfur that's housed in the cell to sulfate. So stage 2 of our technology is designed to create the right conditions for growth of a biofilm containing these sulfur oxidizers. So we start with a tank with a shallow packed bed because we still want an anaerobic environment for the growth of sulfate reducing bacteria so that we can minimize any sulfate produced in the system. The treated water from stage 1 with high levels of sulfide fills the tank, and the surface is inoculated with sulfur oxidizing microorganisms which grow into a white biofilm that's about 2 to 3 millimeters thick. And this is a picture of our biofilm from the lab. It was made up of a complex consortium of bacteria, which we are in the process of analyzing, which were sulfur oxidizers containing granules of sulfur, and we were able to see these under the light microscope. This is not a picture from a light microscope. We also found that a number of elements had precipitated into the biofilm, and then this is the other picture here is an electron micrograph of the biofilm, and it shows that we found struvite crystals as well as crystals of magnesium and calcium in the biofilm. So Another feature of this stage 2 is that the floating biofilm completely covers the surface of the water in the tank. So it prevents losses of hydrogen sulfide gas to the atmosphere and minimizes safety risks from the release of the gas, but it also prevents excess oxygen from dissolving into the water. And this means that we can maintain optimal conditions for sulphide oxidation to sulfur. The film needs to be removed and re-inoculated at regular intervals to ensure continual growth and uptake of sulphide. The aim is to use the floating biofilm to oxidize sulphide to sulfur, and typically, chemically and biologically, in the presence of high levels of oxygen, sulphide rapidly oxidizes to sulphate. But when oxygen levels are low, typically less than 5%, and when sulphide concentrations are high, with a sulphide to oxygen molar ratio of about 10:1, partial oxidation of sulphide occurs to produce elemental sulfur within the biofilm. And these conditions select for the sulfur oxidizers that we want, typically the green and purple sulfur oxidizers, and these guys are the ones who are responsible for production of a biosulfur. We started with proof of concept just to show that we could grow a biofilm using our treated water. And then we used sulfur biofilms from our larger systems, and we did some tests with dried biofilm that was generously donated to us by the Moss Group. And the combination of these two sources of biofilm have formed our inoculant for all subsequent test work. We did batch tests to determine kinetics, and we've done a number of continuous tests just to prove that it works. And next we began integration and optimization studies to reduce the residence time without compromising the biofilm or the water quality. These graphs are from our first continuous tests, which we did to prove the concept that the two stages could be integrated. So you can see here from about day 45, the removal of sulfide starts to increase. And this is because our columns upstream were producing more sulfide. So in this test, we achieved more than 85% removal of the sulfide. It's important to note that there are no water quality guidelines for sulfide for irrigation water. So the aim is to remove as much as possible because any excess sulfide is rapidly converted back to sulphate once it leaves the system. We also measured sulphate to ensure the sulfide wasn't oxidizing back to sulphate. So the system was seeded with sulphate reducers, as I mentioned, to prevent buildup of sulphate in the second stage. This graph shows that the sulphate concentration doesn't change very much within the system, indicating that all sulfide removed was converted to sulfur in the biofilm, and any increases here in sulphate in the treated water entering the system, here and here, were also removed. Now, the mass of sulfide removed and the sulfur produced needs to be quantified, and we will do that in more detail when we scale up the process. So next, we integrated the two stages to form one smooth treatment process and to optimize the parameters to ensure we obtain irrigation quality water. And this is a picture of our setup at Mintek. It's still relatively small scale, treating only up to 6 liters per day, and work is currently underway in preparation for scale up for further piloting activities. And you'll notice in this picture, this is the new biofilm forming. It actually looks like cling film, but this is the start of a sulfur biofilm. So this is some of the preliminary data from our integrated process. We reduced 95% with a marginal increase in the second stage to give an overall removal of 94%. So we expected higher sulfate removal, so these numbers show that the system wasn't fully at steady state yet. We removed 77% of the sulfide, and our final pH was 7.28. The rest of the parameters in the table were measured to allow us to calculate the sodium absorption ratio for the treated water, which is an important water quality parameter for irrigation water. We also removed large amounts of metals in the water through the integrated process, and just a note on the iron concentration there, that in previous runs we actually removed quite a lot more iron, so I'm not sure what happened here. But these numbers for metals removal and for changes in other water quality parameters are only meaningful quality targets for their intended use, which is irrigation. So we compared the data with both the old 1998 DWAF water quality guidelines for irrigation and the new decision support system assessment and management tool for a more complex assessment of fitness for use over long periods for specific sites. And this decision support system has replaced the old irrigation water quality guidelines. But as you can see from this table, our treated water meets the most stringent targets for each parameter, except for manganese and molybdenum. And you probably already guessed, but we're working on a number of ways to resolve that before we demonstrate the process at scale. So while we've been working hard in the lab, we've also been working on tweaks to the process to make it more sustainable over the long term with regard to the degrading pack bed, substrate delivery, removal of manganese, harvesting of the biofilm, and a lot more. We've been on a quest to find partners and funding as well. So we've been very fortunate to receive funding from and to partner with Anglo American. We are relocating our pilot plant to run further piloting studies to test all of these tweaks that I mentioned at one of their rehabilitated mine sites. Since the process was so efficient, we only need single-stage reactors, so we'll be able to run more piloting tests in parallel. We were also awarded funding from Coltech to further optimize the integrated process on site, and this is the pilot scale tank for the second stage. We also received additional funding from Mintek for optimization and piloting of a new strategy for substrate replenishment, including waste materials such as cow manure, shown in this slide. The new strategy should increase the stability and sustainability of the process while reducing the overall operational costs. So some of the bigger players in industry and government, as Tamsyn mentioned on Tuesday, are starting to see the importance of supporting projects that focus on real sustainable solutions. The Green Engine is one of these, and it's being driven by Anglo American. The idea is to promote local business activities and the creation of new industrial hubs in the place of mining once those mines have closed. They're beginning some pilot scale projects this year towards implementation of the Green Engine at one of their rehabilitated mine sites. And we are also partnering with Anglo American to demonstrate the integration of closure with irrigated agriculture as part of the Green Engine rollout. Mintek, together with our mining partner Anglo American, have formed a consortium which includes the Moss Group and the University of Pretoria. We were awarded funding from the Technology Innovation Agency to construct, commission, and operate a demonstration plant, which would look like that. So the demonstration scale plant entails building a number of modules which will treat a total of 50 cubes per day. And to scale up to treat the total volume of AMD produced at that site, more modules like this one would be added in parallel. The water will be neutralized, treated in the first stage to remove sulphate and metals and to increase the pH of the water. And then in stage 2, sulphide will be removed and the water will be polished to produce irrigation quality water, as well as the recovery of sulfur and a biosulfur product. And this project will also include plot trials to use the treated water for irrigation on site. So I hope now you will agree that the closure process is a potentially suitable South African solution for those hard to reach locations, such as those mentioned for the Olifants Catchment. It is a low-cost technology. It has low to no energy requirements. It's low maintenance and can be considered a low-tech solution. It's sustainable over the long term, and it's particularly applicable for sites that aren't suitable for the current treatment alternatives, so it's intended for sites with low volumes in remote locations and in areas where there are a lack of services or infrastructure. So with a sustainable solution in mind, the end goal of the closure technology is to produce treated water that's fit for use in irrigated agriculture, and the water can then potentially be used to produce commercially valuable products, including food, forage, cut flowers, and energy crops. There is an added benefit that biosulfur could be recovered either as a saleable product or reused as a fertilizer. Once demonstrated, we think it would become an appropriate alternative that we can include in our arsenal of sustainable solutions for treatment of acid mine drainage. So in conclusion, let's imagine the possibilities. This is an actual picture of a rehabilitated mine in Mpumalanga after a coal mine closed. So imagine, if you will, that these graded areas in the background are ponds of a closure treatment plant for producing irrigation quality water for reuse in agriculture. 1 megaliter of treated acid mine drainage can irrigate around 35 hectares of farmland. And this can generate opportunities to promote agri-industrial hubs, grow microeconomies, provide employment and food security in an agriculturally important catchment area. It could change the post-coal mining landscape. I'd like to thank the SAIMM for hosting our webinar, and I would like to thank all of you for taking the time to join us for this two-part series.
Okay, many thanks, Kerri, for the impressive or inspiring presentation. Ladies and gentlemen, any questions from your side? I don't see any questions under the Q&A, and I also don't see any questions under the chat. Okay, so in the meantime, of course, this is a topic I'm very passionate about. I do have a question. Kerri and Tamsyn,
Yes.
in the initial step, you do mention a neutralization where the pH is increased. Have you, by any chance, considered using waste such as coal fly ash for the neutralization? For me, this sounds like then it will be quite elegant because this CFA is quite close. Is in relatively close proximity to the acid mine drainage. And then also the resulting sludge can be subsequently dried and possibly used, and thereby achieving a almost zero waste process.
Yes, I would agree with you. I think it's actually been studied quite a lot using coal fly ash as an alternative for neutralization. So it is something that we had considered, but we do have a few concerns. And one of those is that the metals in the coal fly ash might leach more metals into the neutralized water and provide an additional waste stream that we would then have to process. So it's something that we have thought about. It is something we may look at when we get to the demonstration scale. But at this stage, our favorite process really is bioneutralization, similar to the Anglo-American plant that I mentioned in the one slide, because this wouldn't produce any waste at all, and it would use just bacteria. So like I say, there are a range of options that we are considering, and the coal fly ash is quite an elegant solution, but there are some downsides to it that we also have to take into account.
Okay, many thanks for that answer. And then on the very same theme of going for zero waste valorization, Queen has a question here that asks, South Africa has extensive discard coal dumps consisting of coal fines with high ash content. These are significant environmental hazards themselves. Do you think it will be feasible to repurpose this material as a bed substrate in the closure process?
To be honest, it's not something I had considered. I don't know if Tamsyn wants to jump in here, but it's not something we had considered. So the main reason that we need a bed material in our sulfate reduction stage is we need a support so that the bacteria have somewhere to sit. Otherwise, they would just wash out of the system. And sulfate reducers like to form biofilms. So the main purpose of the packed bed is to house those biofilms. And typically, in other examples, people use an organic bed so that you can provide a food source to those microbes. If we were to use something like the waste from the coal dumps, there would be no organic material in there that could feed food to the microbes. And I'm also not sure that the constituents of the mine dumps, if there would be a suitable substrate to actually be able to grow the microbes on in the first place. But like I say, it's not something that I had considered, and I think I'm going to go and read up a little bit about that. I don't know, Tamsyn, if you have any further inputs.
I think I agree with the comments that you've made. Something else that just jumped into my mind is the possible hydraulic issues of using fines, which would definitely be an issue as well. But yeah, it isn't something that we have thought about before, so can go and read up. Thanks, Quinn.
I've made a note.
Okay, many thanks, ladies. Another follow-up question then from Queen. Has there been any work done on the genomics of sulfate-reducing bacteria? And do you think that could offer some insights into optimizing these processes in the future?
Absolutely, it would offer quite a lot of insights. We have a project at Mintek now. It's a science boat project, and we are actually starting to characterize all the different microorganisms that we have in both stages of the process. So we are quite excited about the piloting activities because I think it'll give us quite a nice sample, and it will show us how the microbes change as we change process parameters. There has been quite a lot of work done in various systems, but the makeup of your microbes is very dependent on your feed, on the feed to your reactor. So it's dependent on which organic materials you would feed it. And remember, there's a complex consortium of microbes in there. It's not just sulfate reducers. You've got so many other groups that do hydrolysis of your substrate, that produce acids, that produce volatile fatty acids. It's enormously complicated. And so depending on what you feed, depends on how that consortium, what that consortium will look like. And the other thing is, it depends on what your acid mine drainage looks like, because certain microbes might not be able to tolerate certain conditions, whereas others would thrive. So as you use a different stream of acid mine drainage, or as you feed a different substrate, you get quite a different, quite big changes, I think, in your consortium. So yes, we are currently analyzing and characterizing the microbes that we have growing at Mintek, and from samples from our old pilot plant, and we will be doing this at every stage as we scale up to see how the process actually impacts our microbes. But just another point, it's very interesting, and it is actually useful to know, but these systems are outside in the environment. So they're not using pure cultures, they're not sterile, and any microbes that live in the environment, you know, from the soil next door, or that get blown in by the wind, if they can exist in our process, and if they make it better, then we want them there. So even over time, over say 10 years with the same conditions, you might see quite a variable, you might see varying changes in on your consortia.
Yeah, many thanks, Kerri. So talking about the different stages, I have a question from Paul. What is the schedule for the demonstration plan? So start, duration, etc. Could you please take us through?
Well, I will say this, lockdown has impacted that schedule somewhat. We were awarded our funding from the Technology Innovation Agency just before lockdown. So at the moment, we're busy with the admin, because that's what we can do during lockdown, and planning and drawing and designing and, you know, all of those exciting things that you do beforehand, not to mention the Mine Health and Safety Act and inductions and that kind of thing. So we've been busy with that for now. The schedule for the project is that the plants will begin construction as soon as lockdown is finished, and the same for our piloting activities. We will start up the reactors as soon as lockdown is over, and then hopefully next year we will be able to start operating our first stage, and we'll be able to start generating data at that scale.
Oh, okay. And then another question. Then, how long do you run these tests for? Your typical demo plant?
So the demonstration plant, we hope, will just run until there's no more acid mine drainage. So the idea is that we start with like a module to test, you know, and to sort of get used to operating it in that environment. And then once we have that right, we will add additional modules so that we can scale it up, and we would be able to treat all of the acid mine drainage being produced at that particular site. And as I mentioned, it's intended to become part of the rollout of the green engine at that site. So for this particular plant, we don't want to see an end. We actually want to see it become, you know, a long-term solution at that site. For our piloting activities, most of the piloting activities in literature, and also what we did on site, they were about 18 months to 2 years. So that's why I say we needed to think a little bit about the long-term sustainability of the process. So the new piloting activities, I think, will run for longer than 2 years. Yeah, just to prove that our tweaks are working, if you like. Tamsyn, okay. I don't know if you have any more inputs there.
No, I agree with everything.
Okay, many thanks for that comprehensive answer. Now there's a follow-up question from my side, because then from the presentations, both today and earlier in the week, you keep on mentioning low volumes. So for me, I'm a little bit worried about the scale, because of course this water is targeted for irrigation. And Tamsyn did mention earlier in the week that irrigation consumes a whole lot of water. So then if you're only saying you are targeting low volumes, and yet we know there's lots of AMD in South Africa, and it's also targeted for irrigation, shouldn't we be looking for higher volumes instead?
So Yes, you are right. And as Tamsyn mentioned on Tuesday, that we do need a range of solutions because the acid mine drainage decant points are variable. The water quality is variable. The volumes coming out of there are variable. I think 4 megaliters of acid mine drainage. Just correct me if I'm wrong, Tamsyn. And so there are sites that produce more acid mine drainage than that, and obviously there would need to be other solutions. So one of the difficulties with a process like this, a passive process, a passive biological process, is that it's a lot slower, which means it needs quite a lot more surface area to be able to handle the high volumes, which is fine if you have endless amounts of land, but it's not fine if you also want to have farmland on the same site. So that's kind of the trade-off that we have. And then what was the
if I can jump in here, Kerri?
Yes, please.
So the one of the things that we have thought about is obviously if we've got too much water after the treatment process, what do we do? So we have to remember, like as Kerri mentioned, that we will have seasonal variations on the flows and the volumes of the decant. So if we want to be able to have sustainable agriculture, irrigated agriculture throughout the year, we're most likely going to have to put in place some kind of storage dam or something for the treated water. So in the dry months, when the decant is lower than required for irrigation, we can at least tap into our storage facility. So it's going to be a number of, we're going to need to look into a number of options, but that's kind of, we need to look at small point sources, and we need to actually become involved in agriculture and use agricultural specialists to help us put this into place. And that's why we are going to be working with the University of Pretoria and John Annandale, who's been working with irrigation of crops with gypsiferous mine water for many, many, many years, and he's an expert on this. And he will be giving us a lot of input on how we can do this.
Okay, many thanks for that comprehensive answer. So one more question from Queen. Are the bacteria used in closure related in any way to the sulfur-based ecosystems found around the deep sea hydrothermal vents?
They are actually. They are related. The microbes are related. They're not exactly the same kind of microbes because those environments around the deep sea hydrothermal vents are quite extreme environments. They've got amazingly high temperatures, I think in excess of like 400 degrees, if I remember correctly. So we call them extremophiles because they live in salty environments at high temperature and at high pressure. So while they're related and they form some of the same groups, they're not the exact same bugs. But it would be nice if Mintek could maybe subsidize a project where we could go down and collect some from the bottom of the ocean, I think.
Okay, thanks for that answer. I have a question for Kubashini. Can you explain the process if pit lakes are used? So would pumping of water be required?
No, I think it would depend on your example. So the examples that we heard were actually at the IMWA conference about 2 years ago. And how they explained it is that they keep the pit lakes in place because what it does is it creates an anoxic environment at the bottom of the pit lake. And what this does is it prevents any oxygen from going into the areas where the pyritic ores are, which prevents production of acid mine drainage in the first place. It's not completely foolproof. So the pit lakes, at least the ones that we saw at the conference, they tend to overflow, and there might be a small little treatment plant to make sure that there's no contamination of metals into the environment. But in these cases, what they did is they injected ethanol, and the ethanol then provides an organic substrate for the microbes, for the sulphate reducers that are sitting down in those anoxic areas. So if there is any acid mine drainage decant, such as in times of this high rainfall and the groundwater level rises or something like that, then those sulphate reducers will just keep the contamination of metals to a minimum. And then any water that overflows into the environment is considered a suitable quality. I hope that answered your question.
Okay, many thanks. Yes, that is quite comprehensive. Paul has another question. What level of routine maintenance is required for a passive process such as this one?
To be honest, I think that's something that we will work out once we get to the demonstration scale. So at the moment for our pilot plant, most of the routine maintenance is around collecting samples and analyzing samples. I had a really nice slide which showed all of the hard work that we put in on site, but the presentation became a little bit too long. But most of it is in managing the pumps and managing flow rates and those kind of things, which you need to do for a small scale system where you are trying to collect good scientific data. So I think, if I remember correctly from the other examples in South Africa, you probably need somebody just to check on things every now and again and maybe take a sample once a week. So it really is minimal maintenance requirements. And then depending on the solids buildup in the bed from the organic substrates or from the metal precipitates, you might need to do an overhaul of your bed every few years, which again is what they've done in the other examples. So like I mentioned, we are trying to tweak that so that we can reduce those maintenance requirements and so that we will never have to do an overhaul of the bed. But I think we really will refine that and know how often we have to be on site and how much maintenance we would have to do once we get to demonstration scale.
Okay, many thanks for the comprehensive answer. So there are no more questions. So ladies and gentlemen, we still have we have just over 5 minutes. So more questions are welcomed, but there are also there are quite a few congratulatory messages again for a wonderful presentation. So thanks once again to you, Kerri and Tamsyn, for the great presentations on Tuesday, of course, and also today.
Thank you.
Thank you.
Okay, there are no more questions this side. I'm scrolling through again, so no further questions from the team. So everyone is thanking you for the wonderful presentations. So, and again, all the best for the remainder of the work. I'm of course looking forward then to following up on the pilot plant and on further developments regarding this work.
Thanks so much, Bushler. Thanks, everyone.
Okay, so that concludes our presentation, and thanks also to the SIMM for giving all of us this opportunity. So then next week there will be more webinars, so please tune in then, and have a wonderful weekend, and cheers.